Well, good afternoon, everyone. I wanted to thank you all for joining us. We're very excited for our very first kickoff session of the AHPBA Clinical Trials webinar series. This is an idea that came up through the Clinical Trial Committee at the last AHPBA meeting, and we're very excited that in the short time we've been able to put a year-long course together for you. On the slide, you can see the brief outline for the webinar sessions over the next year. So today we're really doing an introduction to clinical trials and really focusing on the design of the studies and feasibility as the first concept. And then in June, we're going to go to a session really focusing on endpoints and clinical correlatives and sciences as well as biomarkers. In October, we will then focus on statistics, where we will have a really in-depth session on statistics, and then we'll finalize it with sort of discussing like where do these trials are, like really proposed, budgeting, financing, and some of the more essential things to really run a pragmatic and successful clinical trial. This course is really intended for anyone who is interested in clinical trials, and we really wanted to focus specifically on sort of clinical trials that is relevant to the surgeons. As many of you are aware, there's various sort of different courses available, and we really thought that this would be unique to surgeons, specifically within the AHPB space and area. And so without further ado, I will give over the mic to Dr. Kavanagh, who will introduce our first speakers. We're very excited to have you all. Just two small logistical things that we will unmute everyone just to make sure that the speakers have full attention and there's no background noise. But we really would like to be as participant heavy and engaging as possible. So please use the chat to send your questions. We will take a break sessions between the two speakers where we go into more of an in-depth discussions. And then we will even pick out on some of the audience that I will recognize on the names to really engage you all. We really hope that this can be as much of an interactive session as possible. So with that, I'm gonna hand over there to Dr. Kavanagh for the introduction. Thank you.
Thanks, Seth. It's my pleasure to introduce our first speaker, Dr. Eileen O'Reilly. Dr. O'Reilly received her medical degree at Trinity College in Ireland. Her training included a medical oncology fellowship at Memorial Sloan Kettering Cancer Center, where she currently holds the Winthrop Rockefeller Endowed Chair in Medical Oncology, specializing in pancreatic and hepatobiliary cancers. She's extensively involved in research and clinical trials, both at her institution and nationally. She's a co-author or author of over 300 publications. Finally, she serves as a co-chair of the Alliance GI Committee. It's our pleasure to welcome her as our first speaker in our first AHPBA Clinical Trials webinar. Dr. O'Reilly.
Great. Well, thank you very much indeed. Good afternoon, good evening. Real pleasure to be invited to do this with Andy Lowy, and want to pass on firstly thanks and regards from Dr. Yothers, who's unable to be here this evening, and he's very kindly allowed me to use some of his slides, so giving he a lot of credit in this talk. So it's set up as a talk, but please feel free to interrupt. I'll let the moderators guide us when to pause. So firstly, I just need to acknowledge various disclosures and consulting. I don't think any of this directly relates, maybe just at the very end, in terms of one of the trials to our discussion today. So here's the problem, and I think this is a nice study that many of the group may be aware of, that I think sets the importance and the opportunities in terms of surgical research. And this was a study that was presented at ASCO in 2021 and recently in a letter format in JAMA Network Open, looking at just a cross-section of clinical trials and looking at what the degree of surgical involvement in these studies were. And the authors identified that a little under 8% of the studies, of over 250,000 studies, involved surgical interventions, and about 8% of those involved hepatic malignancies. And there were a couple of interesting observations, and I think this is a credit to the surgical community in terms of how these studies were set up and completed, in that they had the lowest rates of early discontinuation and the highest odds rates of reporting. So something to be proud of, and I think something to build on in terms of opportunities in surgical clinical trial design. So moving to a brief review of what clinical trials look like, and I realize that the audience here may be a little mixed, so forgive if I'm assuming little, but just to level set for the discussion. So phase 1 study can be a first-in-human combination looking at dosing, safety, pharmacokinetics, so how the drug is handled and metabolized, and pharmacodynamics to look and see what surrogate effects there might be in the body. And typically there's been a traditional progression from phase 1 to phase 2 to phase 3. However, in the last 5 to 10 years, we've seen this change, I would say, fairly significantly. And now drugs are sometimes approved even in phase 1. And so a recent example of that is some of the checkpoint inhibitors in these large 400 patient studies undergo all of their assessment to FDA approval within the same one protocol. And that's sort of paradigm changing in itself. In terms of I would say the bulk of the work that we do, and I'm speaking at the cooperative group level, it's sort of in the phase 2, phase 3 space. So we'll spend a little bit of time discussing that, not really mentioning post-approval studies, phase 4. I think that has not perhaps so much relevance for discussion today. And then a lot happening, which we'll again speak to a little bit towards the end in terms of innovative trial designs and how one can be creative as you think about your study, both in terms of numbers, in terms of the questions being asked, in terms of learning as you go, and in terms of multiple sequential evaluations in parallel. So very briefly here, just a phase 2. This is a typical proof of principle study and usually will include anywhere from 10 to 20 up to 100 plus patients. A classic example is a single arm design, and sometimes that can be relative to historical control. But as you'll see, increasing the role for randomization and randomized phase 2s are very favored from the NCI perspective. And these can be open label, they can be blinded on one side. So single arm means typically the participants are blinded, double blinded, both the participants and the treating team, but there can be multiple layers of blinding, otherwise sometimes pharmacy, etc., depending on what the specific intervention is. And these are, in terms of goals, establishing a safety signal, establishing an efficacy signal, and typically looking at an intervention in a defined and more homogeneous patient population. So here's a list of endpoints, and not to go through them, but just to reference that these are common endpoints in phase 2 and in phase 3 trials, and typically response rate, disease control, disease-free survival, overall survival are ones that are typically used. And we also see this more and more in terms of drug and therapeutic trials, looking at dual endpoints. So for example, response rate and progression-free survival, or progression-free survival and overall survival. And they can be co-primary endpoints. There can actually be sometimes different endpoints in a phase 2, phase 3 study, and we'll discuss an example of that a little bit later. So that's one way of sort of maximizing the win, but you do have some statistical trade-offs when you design studies in that context. So this is courtesy of Dr. Yothers, and just a brief review of statistics. I know you will have a more in-depth discussion at one of the seminars later in the year. So in terms of the two key things that we look at from the error perspective, the type 1 error, or the alpha, is the potential for a false positive. So the adjudication of an efficacy signal when in fact your intervention is not helpful. And that can be either one-sided or two-sided based on your statistical design. A beta, or a type 2 error, is the converse. It's a false negative. It's when you adjudicate that something is inactive and when it really is active. And the power then is the probability of finding efficacy when your intervention is actually helpful. And the power is 1 minus the beta. Sample size determinations are always a discussion, and I imagine we'll come into this when we do go to the questions and answers. And that depends on the size of the disease under study. It depends on your intervention. It's often a trade-off between cost and feasibility, and it requires an understanding of what you anticipate your intervention may do. So sample size is not a straightforward consideration. So looking at some additional phase 2 designs. So the single arm example is when we compare to historical controls. That's one way of looking at it. And you could design as either a 1-stage or a 2-stage. A Simon 2-stage example is a very commonly used one in drug interventions, particularly in a single arm phase 2 design where we've identified some activity, we have an understanding of what the safety signal, and this is a first pass in terms of efficacy, with the stage 2 design conserving the number of patients and requiring an inbuilt efficacy signal before it might succeed to the second stage, and that can then prime whether or not to take it further. So that was very traditionally and still is used a lot, but it does have limitations, and the big one being the historical reference. And so the challenges there are that your historical comparison can change over time. That's interpreted as drift, and that can sometimes have a big impact in terms of outcome, especially when the standard of care is changing. I can't say we have that problem every day in pancreas cancer, but what we certainly see is that the totality of all of the care that's delivered in terms of surgical care, oncology care, gastroenterology, treating cholangitis, treating pain, and the interventions that have come from therapeutics, all of that leads to some improvement in patient outcome over time. And that's, we see that even in periods of 5 to 10 years. The problems, of course, that these can be small studies. They can sometimes lead to an incorrect interpretation. So this non-statistical error type 3 is where you adjudicate that something is effective, but you adjudicate it for the wrong reason. So it's not courtesy of your intervention, it's courtesy of other factors that have happened over time. So multiple arm phase 2s are increasingly common these days, and randomization is an effective way to minimize the bias and to address the confounding factors, and some of which one can know about, but many we don't know. And these can lead to various designs. And the other sort of nice version of a phase 2, which is very interesting, is sometimes difficult to follow in terms of statistically how it's set up, is adaptive designs. And this is learning as you go. And the initial patients included in an adaptive trial design inform how the study may change. And there can be, for example, over time, weighted randomizations to the more effective intervention and less people likely to be accrued to arms that are less promising and ultimately dropped. And again, these are very complex and require a lot of oversight. And I think the people who most understand these study designs are statisticians, but they are a great way to determine whether something is effective in a potentially a more limited number of patients and also quicker, and to test multiple things in parallel. So just summing up in terms of phase 2 designs, very traditionally it was the mid-phase evaluation. We see everything now, as you said, studies or agents or interventions being evaluated in phase 1 directly to phase 3 and not going through this traditional paradigm. Although if there is the phase 2 setting that's changing, that's using randomization and using multiple arms. And as mentioned, the NCI has a very strong interest in this, and this essentially we don't do in the late to mid stage setting, we don't do single arm studies in the cooperative group setting just because of all the concerns that we've alluded to. So giving some examples here, and thanks to Matt Katz, and I know Dr. Lowy is going to pick up on this in another context, but just to illustrate how much we can learn from these types of studies. So just a quick reminder on this. This is the Alliance Borderline Resectable Randomized Phase 2 conducted in pancreas cancer. These data were presented at the GI Cancer Symposium in January of last year. And this study was designed to test essentially 2 experimental arms. There wasn't a direct control arm in this. It was referenced against historical control. And the question is known to many, is adding whether or not SBRT or high dose limited radiation following sequential modified FOLFIRINOX in borderline resectable pancreas cancer impacted outcome. An inbuilt interim analysis, and that's one way of protecting an intervention and protecting patients and all of the other ancillary factors for an inefficacious intervention. So this study was designed to not compare the 2 arms, with the exception that it was a pick the winner design. If both arms met their pre-specified reference point, then there was an option at the end of the study to compare the arms directly. But the way the study was set up was comparing each intervention to historical controls, and the historical control reference here was a 50% 18-month overall survival that was culled from a very careful and systematic review of the historical literature in terms of neoadjuvant studies or studies that have been conducted in the localized pancreas cancer space. And that led to, relatively small study in terms of patient numbers, 62 per arm. You'll see the power here. We typically will see a higher power in a phase 3 design, a little bit lower in a phase 2. And not to go through all the endpoints, but just to comment on all of the endpoints. We mentioned this interim analysis, and the arm would be designated as successful if more than 36 patients were alive at 18 months after randomization. And so these were the outcomes, not to belabor the endpoints here, but it concluded that the systemic therapy alone arm, contrary perhaps to I think the bias in the field at the time, that this would have been the efficacious arm, the radiation arm, and not to get into all the details on the why here, but that did not meet the reference point for adjudicating whether or not it met the efficacy signal. It did not, and hence there wasn't a direct comparison of these 2 arms. You'll see the red numbers here just indicating the reference to the 50%. So chemotherapy met it, radiation did not. And so just moving forward from that, that's an example of one that I think we've learned a lot and will continue to learn more, and is being built upon in various settings. So a couple of minutes on adaptive phase 2 and phase 3 designs. I think these are very attractive considerations and inbuilt into the study design, where you may have a phase 2 component and you'll have a designated pre-specified endpoint. If that's met, then there's seamless continuation of the study to a definitive phase 3, and that can save a huge amount of time. It can save resources and, again, patients. And just to note here that some of these studies can have different endpoints. So sometimes in oncology, therapeutic drug trials will look at response rate as approximate signal, or PFS. And then in a definitive phase 3, use either OS or sometimes, but less commonly, PFS, OS obviously being a robust and reproducible endpoint. So moving to phase 3s, these are definitive proof of efficacy studies, can involve usually several hundred patients, sometimes to several thousand patients, and they are almost always randomized. and there can be a variety of blinding considerations. So depending on the intervention, some interventions, of course, you can't blind. Some are blinded to the participants in the study, and some are blinded to all of the both the participants and the medical team. And these are often with an intent to change the standard of care, to refine the standard of care, or to lead to a new drug indication, or to lead to an updating of the NCCN guidelines that would allow a change in practice to be covered. So practice changing studies. And just a brief look at some of the phase 3 designs. We talked about randomization. Often will control for various factors in terms of geography, in terms of performance status, in terms of whether or not somebody had had prior surgery, etc., in terms of stratification factors to try to lead as balanced a randomization in both arms, with or without blinding and placebos. We talked about definitive endpoints. These are reproducible, disease-free survival, progression-free survival, overall survival. In these types of studies, the type 1 error is limited, and the power is generally higher, and that you always want to err in favor of not inadvertently detecting an efficacy signal. So for most phase 3s, we'll see power of 90%, occasionally in the 80s, and very occasionally lower than that. They do typically require data and safety monitoring committee oversight, and they are major interventions from all the criteria that we think about. So a couple of just examples here that have, I think, been very informative in terms of learning. So this is the Agilent study that has defined the standard of care with modified FOLFIRINOX in pancreas cancer. In many regards, this was a very simple trial design. So this looked at patients who had undergone resection, who had recovered from surgery, had a good performance status. CA19 line was within 2.5 times the other limit of normal, and a direct randomization to the prior standard of gemcitabine and modified FOLFIRINOX. And these were the primary endpoints of this study. Disease-free survival was the primary endpoint. It's survival that sort of has caught our eye, but that was actually how the study was designed, recognizing that what we do at the time of recurrence is likely to impact survival, but still the point being that what you do first has the greatest impact in terms of long-term survival. So this study had an 80% power, so it was a little bit on the low side by typical phase 3s, and that helped with getting a reasonable sample size. And you'll see that the statistical design called for this increase in survival over 3 years from the disease-free perspective. This study was clearly positive. And this is an example of the disease-free survival. So it's a hazard ratio of 0.58, which is one of the lowest, i.e. More favorable hazard ratios that we've seen in pancreas cancer. And that, I think, has stood the test. And these data were updated most recently. So another example, which has been, I think, challenging for the field to absorb and to understand how it should be integrated. But nonetheless, as we'll see, this, I think, has led to some follow-on studies, which will certainly impact practice. And this arguably has in Europe. So this is the Prio study, and this is looking at comparing neoadjuvant chemoradiation followed by surgery and followed by adjuvant gemcitabine using a gemcitabine-based radiation approach. And the standard at the time, surgery followed by adjuvant gemcitabine. So first thing to say, these studies are very hard comparing two modalities in terms of, even though it's a timing question, it's randomizing patients to either surgery first versus surgery later. And the way this was designed was to look at a median improvement in overall survival from 11 to 17 months. So looking at this in terms of the updated data that were presented at the last ASCO meeting, this actually did meet statistical significance in the final update. It did not in its initial update. And if you look at the 5-year overall survival, it sort of becomes meaningful looking at the median overall survival. It's hard to appreciate that this actually might translate, but over time it does. And it looks like most of the benefit, and maybe we can come to this in the discussion, may come from the impact in the borderline resectable population. At least on the initial analysis, there was not an obvious benefit to the overall survival in the resectable patient population. So just to comment on a couple of points on this. So I think it's an impressive feat that this has been done and completed. And that's the first thing to say, warts and all here. It's a prospective randomized trial. It was multi-center. Some of the challenges, of course, on this study is that it included 2 patient population, and you can argue about whether that's good or not. The fact that the standard of care changed over time, and this is very clear that this happened during the conduct of this study, and that in most people's perspective, at least from the US and Canada, neoadjuvant single agent gemcitabine and chemoradiation would not be considered a go-to in the neoadjuvant space. And the big question that we end up drawing sort of puzzled conclusions is, how do we move forward with this? And to the credit of our colleagues in Europe, they have designed this study, and not just designed this, now completed this study. And I think this is very impressive. And this is sort of building on what was learned in that first trial. So the winning arm, which was, by all accounts, the chemoradiation arm, and comparing neoadjuvant FOLFIRINOX, which sort of gets us to where the field is today. And this completed recruitment about a year ago and is maturing out. Again, reasonable sized phase 3 and powered here for overall survival. So I think these results, as they read out, will be very informative. And these 2 studies, a shout out here. So acknowledging the Prio Plank 3, so this is an iteration of the Dutch pancreas cancer group, 2 very similar studies. One will be conducted in Europe, the Prio Plank 3, and A021806 in the US, looking at neoadjuvant or perioperative therapy compared to surgery first, followed by adjuvant treatment. And this is led by Christina Ferroni and Akil Chawla. And so just a quick look here at the statistical design on this. And this was powered for looking at 88% power here, looking at a superiority design on the perioperative arm and basing the control arm on the previous studies that we had to reference what neoadjuvant therapy does in the preoperative space. So the guesstimates here from surgery and adjuvant therapy, 2-year overall survival of 37%. So recognizing here the clock starts at the time of enrollment. And so that means that patient has to go to surgery, recover from surgery, be well enough to start adjuvant therapy and receive adjuvant therapy. And that's why that number seems very low compared to what we saw in the FOLFIRINOX study. And then the projected goal with the intervention arm, the perioperative arm, is to move that bar to 53%. So this study is well underway, and maybe our colleagues on the call will add a little to the discussion afterwards. But I think it's a phase 3 question, superiority design. But either way, this will be informative. And I think it's very likely that we're going to conclude that it's not going to be one approach is suitable for all patients, but we'll have some additional subsets to further understand. So for the last couple of minutes, just a couple of comments on biomarker designs. And I know this will be a topic again of further discussion. And I think this is of great interest, and biomarker can be everything from a mutation to a strategy for enrichment for a population at risk or likely to benefit from an intervention. And these are very favorably looked upon. And from the NCI perspective, they may be integral, as in key to the primary endpoint. So, for example, BRAF-directed therapy requiring BRAF mutation testing, or MSI directed therapy being another example of an integral biomarker. Integrated are hypothesis validating, but not essential to the primary endpoint determination. And perhaps the practical consideration of this is where the NCI may support some of these endpoints. So just very briefly to give an example of, I realize we're talking about surgical-based trials, but this is an example of a biomarker-enriched study that I think was creatively designed and has established a number of things, established a value to targeted therapy in advanced pancreas cancer, establishes the value of a maintenance treatment, and looking at a difference in outcome of progression-free survival in a relatively small number of patients. And this is, I think, precedent setting, not so much in terms of the win for the intervention, but the fact that it's established a new paradigm to build upon of this concept of maintenance space in pancreas cancer. You'll notice here just a very, very large number of patients that were screened. This was a positive study, but where I want to go is the learnings from that study have now brought this forward to an adjuvant trial that's being led by our colleagues in ECOG. That's a very simple 2-to-1 randomization of olaparib versus placebo. This is designed as a definitive study. It's not designed currently with registration intent, but designed as a definitive randomized phase 2. And if this were positive, this would lead to inclusion in the NCCN guidelines. And actually that would be sufficient to integrate this as part of standard practice. So last moment or two just on emerging platforms and new trial designs. And just to give you an example of what a basket study looks like, we do these a lot in biomarker selected populations in various diseases. This is one mutation, multiple common diseases that it may occur in, and a single intervention and parallel evaluation. An umbrella study design is a single histology and counterpart and multiple interventions in that single histology in a controlled way. An example, a very current example, and again, I suspect a number of the audience are familiar with this, is Precision Promise, and both Dr. Lowy and I have direct involvement in this, and I think this is really interesting. It's in the advanced disease space, but hopefully the next versions will bring it forward to earlier stage disease and localized and ultimately resectable pancreas cancer. And so again, designed to evaluate multiple interventions in parallel using contemporaneous control arms. So your control arm is always sort of up to date and current. Designed to look at biomarker enrichment strategies, hasn't as yet tested that exact hypothesis, but various opportunities for surrogate endpoint evaluation and a whole series of supportive care interventions. And this is designed within the context of 175 patients of stage 1, 100 patients, then an additional 75 patients could actually lead to a drug approval in pancreas cancer based on the way this is designed. Again, we don't have an example of yet if that's come all the way true, but there are a number of very exciting arms in this. And related to this in the UK, we have the Precision Panc version of Precision Promise. And just I use it here just to show the schematic, how trials can be in the preclinical space, in the discovery space, and of course in the clinical arena. And so that's where we hope to see Precision Promise go, just earlier stage disease, but even more preclinical testing and sort of setting up the platform. So for those who may not have seen this, this is a nice paper in the Journal of Thoracic Oncology looking at some of the principles of clinical trial design. And Dr. Lowy is going to expand on this in more detail. And I think first and foremost is trying to identify that question and to be able to ask the question and to make sure that it's a clinically meaningful and important question. And this takes a lot of discussion, I think a lot of time, and often a lot of back and forth and iteration. I think that's, and refinement, I think that's so true. And all of the studies that we see in the cooperative group level have often gone through a year, 2, sometimes even 3 years of discussion. And we can argue about the merits of the longer aspect of that, but I think there's certainly huge benefits to the discussion and the feedback that comes up from all of the review processes. So I'll just sum up, and I'm sorry I've taken a few extra minutes here just to try to drill down onto the critically important question, to try to ask that in the most straightforward way that we can. I think there's a huge value to simplicity, and I would say our European colleagues in terms of trial execution have mastered this. Sometimes we try to build too much into studies, and I think we lose sight of what we're trying to answer. Just to bring up this discussion of all of the variables in terms of the study size, the power, the frequency of the disease, the resources and the time, and all of these factor into what the trial ultimately looks like. I think a huge wish, of course, to have some surrogate endpoints and more proximate signals, but to make sure that they are surrogate endpoints that have value and that are appropriate for clinical decision making. And I think 2 last points here, that to make it our mission to publish negative studies, and we learn a lot from negative studies, and that's a key thing, and just to recognize that there's no perfect right or wrong way to do this. There's a lot of very reasonable ways to do this, and I think that's the process of discussion and trial design. So thank you very much. I'll stop there and pass it back to Dr. Kavnar and Dr. Galami.
Thank you, Dr. O'Reilly. What a fantastic review. We will just go to our next speaker. Dr. Kavnar will do the introduction, and then we will have a good 15 minutes at the end so that we can avoid some overlap and have a really in-depth discussion at the end of the session.
Thanks, Seb. Dr. Andrew Lowy is a professor of surgery at the University of California, San Diego in the Moores Cancer Center. He's a surgical oncologist specializing in pancreas, liver, and GI tract cancers. He's been a pioneer in surgical treatment of pancreatic cancer and peritoneal surface diseases. He's a funded investigator holding multiple R01 grants related to pancreatic cancer and has published extensively. He's an experienced clinical trialist and is involved nationally at multiple levels, including his role as a co-chair of the NCI Pancreatic Cancer Task Force and as a co-chair of the Swog Pancreatic Subcommittee. It's our pleasure to have him here as our second speaker this evening. Dr. Lowy?
Well, thanks for the kind introduction, and good evening, good afternoon, everyone. I'm delighted to chat with you and follow Eileen, who's always a hard act to follow. I'm going to probably overlap a little bit with some of the comments Eileen made, and to kind of stress some of the really important things that she pointed out, but then focus a little bit more about designing trials and and what goes into that. And the first point I really wanted to make, which I think all of you probably recognize, but I think it can't be overemphasized, about how difficult clinical cancer research really is. And I always point this out, to trainees, that because we always think about folks in the lab and how smart they are and doing lots of things that many of us don't always understand at the molecular level. But honestly, from my perspective, clinical research is actually much more difficult from the standpoint that it depends fundamentally on people. And whenever you have numerous layers of humans involved, there are numerous layers where things can go wrong for many reasons. And so I think the point that Eileen stressed about how well our European colleagues have performed clinical trials goes to the fact, again, that they have simplified things as much as possible with respect to questions. Because at the end of the day, as I say in the last bullet point, it's not only challenging to conduct a trial that ends up with a positive outcome, meeting the hypothesis that was designed at the beginning of the study, but it's challenging just to get the study completed. And so there are many, many barriers to that, and simplicity is really your friend when you're trying to reach the goal of getting your trial done and getting an answer, whether it be positive or negative. So just something to always, I think, keep in the forefront of your mind as you're embarking on leading clinical research. So, the good news about our field is that for a very long time, whether you're talking about pancreas cancer, hepatocellular cancer, biliary tract cancer, we really had very few options as far as designing new studies, particularly in the setting of new drug development. And we all know, and this is by now a fairly famous figure from Lola Rahib's paper in Cancer Research in 2014, where that was written to demonstrate the increasing incidence of pancreas cancer. But important to note on this that the incidence of all HPB cancers is actually rising. And I think everyone in the AHPBA is familiar with this problem. And while that's a bad thing, the great thing is that science has brought us a greater understanding of the genetics and the molecular drivers of these tumors. And there has been an explosion in drug development. And so we have the opportunity now to conduct more exciting and many more studies in this space. That's great, but it comes with challenges because on the whole, these are still relatively rarer cancers. And as we start to parse them into molecular subgroups, as Eileen talked about, we get smaller and smaller cohorts of patients to study, and that makes clinical trials even a bit more challenging. So it's even more important that as we think about designing trials, that they are both efficient and they effectively ask important questions. So how can we do this better? Because I would argue that certainly in the past, certainly in the pancreas cancer space from years ago, we made some often critical mistakes as we moved along the continuum from preclinical space to getting trials done that result in positive outcomes. And so I think several important, key things to keep in mind as we design trials. First, we want to ask important questions, and we have to create designs that make answering those questions feasible. And you'll hear over and over again us talking about feasibility, because again, if you don't get the trial done, you don't get an answer. We want to test scientific hypotheses where the preclinical data is very robust, or that we have prior phase data, i.e. the phase 2 trials that Eileen was talking about, or compelling responses even in the phase 1 setting, which we now see more often in the setting of molecularly targeted therapies. It's really critical and something that we have not done well at all in the past, but that happily we are learning, is to try and marry science to every trial, to have some type of way, either whether it be via tissue, whether it be blood-based biomarkers, to try to understand what has happened in the study. Eileen mentioned that we can learn a lot from negative trials, and that's absolutely the truth and really should be the standard that we can learn from negative trials. But I'll show you an example in a little bit where we even had a positive trial, but yet we couldn't really use those results very meaningfully because we didn't really understand what was going on. We want to design trials where the bar is high for moving to the next phase or to create an approval or change in practice. So we don't want to create trials where we'd be happy with a hazard ratio of 0.9 or where we move the median survival by a couple of months. In rare cancers in particular, patient resources are so incredibly precious that we really want to enroll patients on trials which are going to make really big, impactful changes to our standards of care. Eileen talked about the use of surrogate endpoints and adaptive designs. which I think are really becoming more the standard, than the exception, with good reason, because they allow for efficiency, to decrease accrual needs and allow us to accrue oftentimes more rapidly. So as many of you on this webinar are aspiring trialists, I just want you to be assured that there are an endless number of important questions to be answered and for you to help us advance our field. And I just listed a few off the top of my head, focusing specifically on pancreas cancer. I think we saw the fact that neoadjuvant therapy is now being investigated in the trial being run by Christina Ferroni and the Prio PANC3 study. We also have a question about how much neoadjuvant therapy is enough. We don't really know the answer to that. We don't know if we need to give patients 6 months of therapy, 4 months of therapy. Should it be guided by some measure of response? Unclear. We still, amazingly, do not know if there's a role or what the role is for radiation therapy in this disease. Many of you are faced with your outlier patients who have a single focus of metastatic disease, who've been doing well for a long time on treatment, and the book says don't even talk about doing surgery on these patients. We're in a different era now, and I think it's reasonable to ask the question, is local therapy, whether it be resection or radiotherapy, is it ever helpful in patients with metastatic disease? We have in the cooperative group setting some proposals about ctDNA and how to evaluate whether it is a useful tool to monitor response, relapse, tell us about whether we should change therapies. And the question about switching chemotherapy, again, comes up in that context and others. We don't understand if we should keep patients on chemotherapy when they have an established, stabilized disease in the metastatic setting, or is actually some type of maintenance therapy, could that potentially be a benefit in patients who have had resection and chemotherapy? We don't know the answer to this question. We know that there's basic science and lots of evidence that drug penetration can be an issue in pancreas cancer, but we don't really understand how much of an issue it is for efficacy. And I would say that a major hole in our field is understanding how and which preclinical models might parallel the responses we see in patients. This is a minor partial list of one disease that we all treat. And so I would put it to you, the optimistic view of all of this is there's a lot of work to be done. And so we need all of your minds to help us address these and many other important questions. So this slide reflects what I was chatting about before, and I forgot to put the reference on, but this is a graph of the survival curve from a trial conducted by Malcolm Moore for the National Cancer Institute of Canada some years ago, which was investigating patients with stage 4 pancreas cancer and comparing their survival when treated with gemcitabine, which was the standard of care, compared to the addition of erlotinib, an oral EGF receptor tyrosine kinase inhibitor. And the outcome of this study was that it met its primary endpoint, which was an improvement in overall survival at 1 year for the experimental arm, which was the erlotinib arm. And this trial has been roundly discussed and criticized because the difference in the median overall survivals was only about 2 to 3 weeks. And yet this led to the approval of erlotinib, both in the United States and in Canada. The problem was that with this small incremental benefit, and with the additional toxicity that erlotinib added to patients, causing diarrhea, rash, the question about, was it really worth it to add this therapy? And so it really never gained very widespread use. And on subsequent trials evaluating it in the locally advanced setting, in the resectable setting, really never showed much of a benefit to erlotinib. And so what I would say about, what I would hypothesize about this study is there probably was a small subset of patients who received erlotinib who got a tremendous amount of benefit and probably drove the small incremental separation of the curves that you see here. But there was no science really embedded in this trial. we never could really understand who was benefiting from erlotinib. And so this drug, which is still on the NCCN compendium, really is an orphan in the wilderness. And we really never understood even a positive trial in pancreas cancer, so rare, but it really ended up being more of a dead end because we couldn't understand the results. And so that's even more important, obviously, when we have a negative trial, you could argue. And so if we don't embed science, if we don't have a real understanding of what's going on or a way to ask those questions, it's really hard for us to meaningfully move forward. So I think this is an instructive, historical trial to look at and think about. So let's move to talking about what everybody's really most interested in, which is, how do you get started when you're interested in being involved or leading a clinical trial? Well, I think Eileen alluded to this. The most important thing is you start with your question. What is the question that you are interested in studying? And to understand whether it's actually makes sense to study this question, you have to ask the hard question about, is there really sufficiently strong preclinical or clinical data to underpin the question that you want to ask? And so with respect to a very early phase trial, you want to understand, is the preclinical science relevant to the clinical question? And this is an issue that we often have a pothole we've often fallen into, where we look at a preclinical study and we then design a clinical trial, but we've designed the trial in a way that's very different than the way the preclinical studies were conducted. And so we have to be very careful about that, because that can obviously change. So if we do a preclinical study and it's done in a primary tumor within the pancreas, for instance, and then we want to apply it to metastatic disease, maybe that preclinical model doesn't really mirror the same setting that we are studying in patients. And so you want preclinical science that's going to mirror your clinical question, and you want it to be robust. Ideally, that it wasn't one study. Ideally, it's been conducted in different ways with orthogonally examined outcomes by different groups, ideally. If it's a more advanced phase trial, you want to make sure that there was a well-conducted phase 1 or phase 2. And if it's a phase 2, because you're asking a phase 3 question, was there really a convincing safety and efficacy signal in that study? And one of the things that we've unfortunately done in the past, especially in the pancreas cancer space, is we will look at phase 2 data and kind of turn our heads to the side and squint and try to convince ourselves that the outcome, that the efficacy signal is strong enough. And in some instances, we have gone on to later phase studies when we didn't even really meet the phase 2 endpoint, believe it or not. And not surprisingly, when we do that, we usually end up with a negative phase 3 trial. The next important thing to answer and to ask yourself is, if we do this study, and if our hypothesis is shown to be correct, what happens next? If it's an early phase study, what will be the next question that we ask when we go from there? If it's a later phase study, will a positive trial really change practice? And these are also difficult, but really important questions to ask. And I'll talk about it later, but these are the questions that you will always get if you are presenting a study to an NCI task force, and it will be asked time and time over and over again at the level of the GI steering committee that has to approve that trial to be able to open in one of the NCTN groups. Equally important, feasibility. Can we answer this question in a relevant time frame? And to answer that, we have to ask the question, do we understand the patient population? Is it well defined? Are there enough patients to accrue? We all, as investigators, have rose-colored glasses whenever we are thinking about accrual. We all think we're going to be able to accrue very easily. That's never going to be a problem, but it's always a problem. It's always an issue with rare exception. And so we have to be very, very honest about this. We have to consider the landscape of trials within our own country, our own regions, as well as the questions that are being asked about by our European, Canadian colleagues, Asian colleagues, what questions are being asked elsewhere in the world to make it relevant, because if it takes us 5 years to answer, to accrue to the trial, and someone else is going to be done with a trial that's very similar design in 2 years, our question may very well become moot by the time we even finish accrual. And so that goes to the question of competing studies, ongoing planned studies addressing a similar question. Now, sometimes having, it's never a bad thing to have 2 separate trials ask the same question, because obviously one trial is not always definitive that tells us the truth, but we have to consider obviously these other designs. What's the timeliness of them compared to one another? Does it make sense? It's something important to always think about. So now I'm just going to give you a couple of examples, kind of like Eileen did, but I'm going to focus on studies that have been done and what were we thinking about? What were the considerations as these studies were being designed? And so I'm going to go forward first and then go back in time. First, going to talk about Swog 1505, which I think by now most of you have heard about, read about. This was a trial led by Devendra Sohal in Swog, and it was a study that evaluated perioperative chemotherapy for patients with resectable disease. It was a phase 2 randomized clinical trial design. And at the time this study was being considered, one of the major questions in the field, which is still a question in the field, was the role of neoadjuvant or perioperative chemotherapy versus adjuvant therapy. Now, there were 2 effective, as there are currently, systemic regimens validated by phase 3 trials for stage 4 disease. FOLFIRINOX, which had also been validated as an adjuvant therapy, and gem-nab-paclitaxel, which was a regimen validated in stage 4, had not yet been reported on in the resectable setting, but actually in the community has been the most commonly used regimen in advanced disease patients due to its slightly better tolerability. So some of the key questions that Devendra and the team wanted to ask were, number one, in a cooperative group setting, not in a single institution setting, where most of the reports really that were available, what are the outcomes following perioperative chemotherapy? And we wanted to understand in resectable patients, are there substantial differences in outcomes or toxicities between the 2 most commonly used systemic regimens, as a means to then go to the next step about asking more deep questions about either the neoadjuvant versus adjuvant question or the question about other agents in combination. And so these were some of the design considerations for Swog 1505. Well, first of all, what phase study should this be? Well, as we thought about this, we knew that safety data existed for both regimens in the perioperative setting. There was reports on both of these that were fairly extensive. As I said, there was no real perioperative standard of care. We did not have a lot of controlled data, and so we didn't feel like we're quite ready to ask a phase 3 question. And so therefore, we kind of arrived at, we have good safety data, we have some efficacy data, but not in a cooperative group, multi-institutional controlled setting. And therefore, a randomized phase 2 trial seemed like the most appropriate design. We had 2 regimens we wanted to study. We wanted to make sure that there were not biases. And so we wanted balance in the prognostic factors as a critical feature. And we thought about, well, where will it lead? Well, the possible next steps could be a phase 3 trial. It could be a phase 2 trial to add on to the chemotherapy backbone. And the statistical considerations were that there was really no reference standard. We were hopeful that both regimens could be effective. So we were going to pit them against a desired, rapidly determined survival outcome. And so we chose 2-year overall survival as the primary endpoint. And this is the data, the survival curves from the study published in JAMA Oncology. And you can see, as I think most of you know, that the overall survival for both arms was similar, eerily similar, and that if you look at the outcomes for the patients in table 2 on the right, the R0 resection rate in both arms was identical. There was a small difference in complete responses, but again, these are small numbers. And while the median overall survival was slightly better numerically in the nab-paclitaxel arm, that was not statistically significant. And so, so both arms performed similarly. Now, the primary endpoint was the 2-year overall survival with a null hypothesis that of 40% 2-year overall survival. This was this study had very high power for a phase 2 trial. And the idea here was that we would see an increase in the 2-year overall survival to about 57%. Now, both arms outperformed numerically the null hypothesis. There was about a 47 to 48% 2-year overall survival. However, neither of them met what we hoped, our aspirational outcome of a 2-year overall survival of well over 50%. What we learned was that the regimen's toxicities were fairly similar between the two of them. And so the conclusion really, from an outcomes perspective, was that the regimens in the perioperative setting performed similarly, both with respect to activity as well as with respect to toxicity. Now, we learned some other things from this study about the importance of central radiology review and how important that is. Was also an important outcome of the study. And so what has happened since SWOG 1505 was performed? Well, both of the outcomes that I alluded to as far as follow-ons have either taken place or are in the planning stage. And so Eileen showed you Christina Ferroni's and Dr. Chawla's study 21806, which is ongoing, which is now asking the question about perioperative FOLFIRINOX versus an adjuvant approach with FOLFIRINOX given following surgery. Within SWOG, there is plans to conduct a platform type phase 2/phase 3 study where the chemo backbone and chosen to be FOLFIRINOX based on the stage 4 data and adjuvant data, given there was no big differences between FOLFIRINOX and gem-nab-paclitaxel in 1505, to adding novel agents and trying to do all of the chemotherapy in the preoperative setting. This study is in the late planning phases, will likely be presented to the GI Steering Committee in the coming months, but this just shows you how what we learned from 1505 has allowed us to go on in multiple different directions to develop, perioperative and preoperative chemotherapy in the setting of resectable pancreas cancer. So the last topic I'll talk about is the evolution of our study of borderline resectable pancreas cancer. I saw Matt Katz on the call. Matt is the unquestioned leader, in this field, having led the initial study, Alliance 21101, which I'm going to talk about, as well as the follow-on trial, which Eileen talked about. But I thought it would be instructive to go back in time to just understand how this really evolved. And you should all be proud that your organization, AHPBA, really was the leader in developing the concept of studying borderline resectable disease, evolving really from work done by Doug Evans and team at MD Anderson. And we had actually consensus meeting to discuss this, which evolved several different kind of consensus statements. And these were those that to facilitate future clinical trials, we had to come up with a standard definition of borderline resectable disease, that they should be studied separately, at least initially, based on objective CT criteria for resectability, and that it made the most sense to treat these patients with neoadjuvant therapy and in the context of a clinical trial. And that trial eventually was AO21101. So as we thought about this study, we talked about, what the optimal preoperative regimen was. The most appropriate systemic regimen was unclear. At that time, we knew the response rates to gemcitabine was very low in the metastatic setting, and that it was higher for FOLFIRINOX, considerably higher, but that it was more toxic. We did not understand, and I would say we still do not understand, what the role of radiation therapy in the treatment of localized disease is. And we knew that the prior studies, which were small in general, studies of borderline resectable pancreas cancer, were quite difficult to interpret. And there were a number of problems with them, as I've enumerated here. So the design considerations for this study were that there was no reference standard. There were no prior trials in this space, certainly none that had been conducted in the cooperative group setting. And so the question came up as we were talking about this, particularly in the cooperative group setting, and particularly with the various steering committees, that we had a question about logistical feasibility. We had said in our consensus statement that evaluating the imaging was critical, and therefore central review was going to be necessary. We didn't really know if we could accrue to a trial like this. And there was a question of variability in both surgical and radiation quality based on experience in this space. All of this led to the conclusion that a single-arm, small, if you will, pilot, or I would say a single-arm phase 2 trial with a primary endpoint of feasibility, not efficacy, but simply feasibility, to establish whether we could do this, whether we could accrue patients, whether this was a safe strategy, and therefore establish some reference outcome standards to then basically build this part of the field kind of brick by brick. And so this was Matt's study design, where patients got FOLFIRINOX. They got chemoradiation, which was standard course chemoradiation with capecitabine. They had real-time central review of radiologic studies and all modalities, surgery, radiation therapy. There was some quality control. And importantly, this study was limited to select centers with demonstrated high volume experience in treating patients with borderline resectable pancreas cancer. And this was the outcomes, kind of the consort diagram of what happened to these patients. I think we learned a lot, even though this was a small study, and certainly we'll be interested in hearing Matt's comments as one who knows all of this data the best. All the patients initiated FOLFIRINOX, most of them initiated chemoradiation, 68% ultimately were resected. A minority of patients went on to receive chemotherapy after resection. And this was the really exciting thing, I think, because we answered the feasibility question very nicely. In this trial, accrual exceeded the projection, which, as I said, rarely happens, but the trial accrued extremely rapidly. And almost every site enrolled at least one patient, so there was broad participation. Strikingly, there was an incredibly high R0 resection rate, 93%. There was a couple of pathologic complete responses. And so I would say this trial was a outstanding example of thoughtfulness. It took, as Eileen mentioned, this trial, Matt probably remembers better than I do, but I think this took at least 2 years to get this study finally designed and then up and going, but really an important, well-conducted trial to build the field on. And the takeaways were that prospective trials were feasible, that central radiology was critical. We learned that we could resect many of these patients with this strategy, and that we could achieve high rates of margin negative resection. And again, as Eileen already presented, this led to a second trial, which already has been done and reported. And while the outcomes of that trial are, I think, still a little hard for some people to wrap their arms around, again, proving that we can do trials in this space and we can address questions in this patient population. So the last slide I wanted to leave you with is a slide that is courtesy of Carmen Allegra from the NCI. And it is a lesson in how to avoid getting rejected by the NCI steering committee. And it really goes back to a lot of the points that Eileen and I have tried to share with you. Why do trials get rejected when they make it through the NCTN task force, the group, the task force, and now they're at the level of the steering committee, kind of the ultimately approval body? Well, these are the main buckets, and I have another slide later if we want to review it about even more granular bullets beneath these. But limited or unconvincing preclinical or clinical support. So again, going to the idea that the preclinical data or the early clinical data has to be strong and robust. The statistical design not adequate to answer the question. And so that can have a lot of different meanings, but essentially they have to be convinced that the result we get is going to be believable and it is going to adequately address the primary question. Feasibility, don't have to say anything more about that. Basically, they have to believe that we can accrue patients to this study in a timely enough manner. And so we have to have evidence that there are patients out there, or we have to have a track record of accruing in this space to convince them that we're going to be able to do this. Competition or overlapping eligibility criteria. Again, it goes to the question of, is this study going to compete with other studies that we have open and therefore make it unfeasible to accrue one or both studies? Or are there studies that, that are going to be done elsewhere that are going to make the outcomes of this study less important? That goes to the study. When we complete it, when we get the answer, is it actually going to alter practice? Or is the next step going to be clear and feasible? If the answer to any of these are no, you're going to clearly get the no rejected stamp on your trial. And finally, again, does this study replicate other ongoing trials or address a question that is embedded in other ongoing trials such that those trials would make your study under consideration be much less important or render your answer moot because the other studies will be reporting first. So I think keeping these things in mind as you design your study will really keep you on the right path and make it much more likely that you're going to be successful in navigating your IRB, or if it's in the cooperative group setting, the cooperative group. So I'm going to stop there and stop sharing so we can see each other and chat. I'm really delighted to participate and, certainly glad to address questions that you guys may have.
Thank you so much, Dr. Lowy. That was a really fantastic, not just overview, but I really thought thought-provoking and really important for us to see really the thought process that goes into really designing a really well-designed clinical trial and all the challenges. I'm just going to start off with Dr. Katz. I think still you might be on the call. I wanted to really hear from you. What were the take-home messages and sort of the biggest learning experiences for you from your study that you did?
I don't see his name on there, Sam.
Yeah, I think he may have dropped off.
Yeah, I may have maybe was it Shashir?
Yep.
Yeah.
Okay, great. Shashir, if you want to, obviously you are now, so involved with, doing the gallbladder cancer trial. Can you just comment on sort of the biggest challenges that you've seen with sort of the time and the experience that you've had over the last several years?
Yeah, well, again, speaking on the theme of the cooperative group trials, I think Dr. O'Reilly said it perfectly that these can take years. So you definitely have to have persistence and patience. It's not something patience is not something that's inherent to most surgeon personalities. So I've really had to learn that. And I would say cooperation and collaboration and being willing to learn from your colleagues. Again, talking about the theme that's been really kind of emphasized in today's discussion is therapeutic cancer trials. And I've just learned so much from my medical oncology colleagues. So you just have to approach this in a very multidisciplinary, collaborative approach, and really like take it all in and be patient to go through the process. And if you persist, like Kim's Apollo study, which again you see something in the metastatic setting. This is something that I've really been a big proponent of. You see something going on in the metastatic setting, which our medical oncology colleagues are running trial after trial after trial, and then trying to apply them to patients with resectable disease, either in the neoadjuvant or adjuvant, in a concurrent fashion. I think that is key. And you see the Polo trial get done, and then Apollo comes out. Now we're doing an adjuvant. We're doing the similar in biliary tract cancers, but that's been a big learning lesson and just good experience in general.
Great.
Dr. O'Reilly, how much do you see the phase of the trial design change as it goes from a concept to approval? I think, for example, of in all of our alliance calls of listening to Josh Smith torture himself through 1,000 iterations of the watch and wait trial. And, obviously there's an interplay between your power, your feasibility, all of that. Maybe you can comment on that process for us.
Yeah, I think that's a very good question, and perhaps one of the things that we learn a lot as we go, and I think Andy summed it up very nicely in this last slide, is not to be overly ambitious and to really focus the question. And again, I think myself, when we're thinking about designing trials, it's the hardest thing is just to try to pin down, be as precise and as specific as you can, is what are you trying to ask, and in what population can you do that? And we saw a couple of examples, I think, during the talks this evening, but a very illustrative one is just scaling back the borderline resectable project. That was, I think, and somebody will correct me, but I think it started out as almost a phase 3 and got scaled down to a 22 patient feasibility study, but it won the day, right? At the end of the day, it was an extremely important study. It set the scene for a whole landscape of trials that have built upon that. And I would also, echo, what Shishir mentioned. I think patience and persistence are key, and perseverance, right? Advocating for an important point. And that, I think, is really key as well. If we believe, if the field believes this is an important question, it's really worth marshalling support. And I do think people come around with thoughtful insights and presentation of an articulate argument that this is something that should be asked and answered.
Yeah, I'd weigh in on that with total agreement. And, sorry that Matt isn't here, but I can kind of speak a little bit on his behalf, and I'm sure Eileen can too, because I think she was involved in a lot of this. But the question about, does the phase evolve? When we started with the borderline resectable question, we were starting with randomized phase 2 designs. Then we were actually, we might have started with a single arm phase 2, and then we were pushed to a randomized phase 2, and we ended up circling back multiple times until we ended up actually at a what was called a pilot study. That's what NCI wanted to call it, because it's really a single arm phase 2 at the end of the day. It's just a small one. And it really did take a lot of persistence and a lot of collective, work and collaboration. And, I think one of the great things about that study was it wasn't just collaboration in a multidisciplinary fashion. It was a multi-group. It was Alliance and Swog, and all the groups kind of working together and really persisting at the GI steering committee that we thought this was important, and we thought that we really needed to do something to move this question and this part of the field forward. And it took a mind-numbingly long time and I know Matt was extremely frustrated, and I just remember having chats with him and saying, look, we just have to keep at it. We just have to, we will win. We will get it done. But, this is what you have to do. And, and in the defense of, the GI steering committee, I would say most of the time they're right about the decisions they make. Nothing's 100%, but there's a lot of smart people who are experienced, in that on that committee, and a lot of the counsel that they provide is very, very wise, and that you have to think about it and not just get upset when you get a rejection or a pending or suggestions about improving it, but to really take it back and work on it. And so, yeah, persistence is key. Collaboration is key. Completely agree with all those points.
Looks like Dr. Martell has a question.
Hi there. Thanks so much to both speakers for a great talk. I just wanted to perhaps unpack a little bit on the issue of pragmatism and trial design. I think you both sort of alluded to it, but I find we're often sort of torn between designing a study that's very pragmatic in nature and then getting into the weeds a little bit and embedding a lot of perhaps biomarkers and things of the sort. And and then this also goes to the issue of perhaps acceptability of the trial eventually. I'm thinking of BILCAP, for instance, where we have sort of a broad umbrella of cancers that are included, and that has to a certain degree made it more difficult for the field to embrace the results. And so I'm wondering if you could speak to that to a certain degree.
Perhaps I can
Yeah, go ahead.
start, Andy, and take a stab at that.
yeah.
Yeah, I think it goes to the question of feasibility, and I know that came up several times, and it's a hard one, and we see this a lot at the level of the GI steering committee. Is this population actually out there, and can you ask and answer the question in a reasonable time, and is that result still going to be informative when the time has completed? And this is very hard. I think to some degree we're maybe protected a little bit in that we haven't had the huge changes in standard of care, at least in pancreas cancer relative to others that helped. But I think if you, for example, think about HCC and you try to design a trial in HCC now in the first line space, I mean, today you'll have one control arm, tomorrow you'll have another, and next week you'll have a third almost in the last few years. So it's hard. I think the days of huge 1,000-patient studies with a single intervention are, they're not going away. They're still going to be important, but the hope is that surgical trial design is going to be precision medicine too. And I think, increasingly we're going to be carving out niches of patients to study interventions that again get around this concept of feasibility.
Yeah, I think that in some ways, the era that we are in and becoming ever deeper in of molecular subsetting and asking these questions about more precise therapies, in some ways makes it a little easier from the perspective is that we can design trials that are smaller with an expected major impact on hazard ratio, because we understand that if our hypothesis is right, particularly about a certain drug target, for instance, when we get to studies, which we will hopefully soon, on KRAS, G12D inhibitors, our expectation is that that's not going to have a minor impact, on the disease. And while we'll then be subsetting, about 35-40% of pancreas cancer patients, so we're taking only now less than half of the patients, our expectation is that we can look for a major impact. We can ask a simple question with a smaller number of patients and really get at it. So I think your question is very appropriate. It is one of the most frustrating things, and it is always a tightrope walk about this balance between asking the simple question that's making it feasible, but also making it important. That's the art, if you will, of trial design. And that's why iteration is really the rule, not the exception, because finding that sweet spot can be challenging, and you need putting a lot of heads together to get yourself to the right place.
Actually, Dr. Lowy, to that said, I was just thinking about the G12C study on the pancreas abstract that was just
Yeah.
presented at ASCO GI.
Yeah.
To that subset, I mean, if you're looking at a smaller, like, population where it's less than literally like 2% of all pancreas cancer, how do you really think that translates into a phase 3? Yes, the impact is larger, but really the numbers are so small. And I think we still struggle with.
Yeah, so that's somewhat of a regulatory question, actually, because as everybody's probably familiar, drug approvals are now happening without phase 3 trials. It's not uncommon at all anymore. And again, and that's because of the scientific hypotheses are so much stronger that, if you see huge impact, where the curves are separated, not by a pencil thin margin, but by, a huge amount, then, the FDA is going to be much more, permissive of giving an approval on the basis of studies done under those circumstances. So I don't think you necessarily always need a phase 3 trial anymore. The old saying was, you throw a brick out the window, if it goes up, you don't really need a phase 3 trial. Something strong is going on here. And so I think that's, more and more the era, that we're in. And adaptive trial design is allowing for us to get these answers, more quickly. That's really what Precision Promise is based on. These arms will be no greater than 100 patients, and yet they will, have the power to lead to drug approvals. And the smart thing that PanCan did was design this study with the help of the FDA, so that they kind of got already buy-in from the FDA about, if we do this, if we meet this, can this lead to an approval? And so I think that was a very intelligent way to go about it. I don't know what Eileen, might want to weigh in as well.
Yeah, I think in an unmet medical need where you see a response rate of 50% in heavily treated disease, that's higher than you might expect with frontline therapy almost. Right there, it's precedent setting, and that context is one that is viewed upon, I think, potentially favorably, and also the possible of disease agnostic, in that you've validated this target and this drug approach in other diseases, with lung cancer and the 12C story. So I suspect it's quite likely that that's what will happen in pancreatic cancer.
Great. Let's take one more audience question. Andrew Fleming.
Hi. First, I just want to thank you, Dr. Gholami, Dr. Kavner, Dr. O'Reilly, Dr. Lowy. Thank you for putting together this awesome seminar. One question about trial design. So when you're implementing one of these adaptive trial designs, how do you minimize or even eliminate the bias this could introduce while retaining the flexibility of the adaptive design? Are these predetermined changes that you can activate, or are these things that you develop as the trial goes on?
So the first thing I'll say is, I am not a statistician or an expert on Bayesian adaptive design, so I'm going to give that qualifier. But what I can tell you is, I'm not sure what you mean by the biases, but essentially what you're doing is you're using the data. The old-fashioned way is you never look at the data, right? You want to look at the data as infrequently as possible, because you're introducing a bias, if that's what you mean by the bias. Here, you're looking at the data frequently, and you are using the information that's being acquired about whether it be response, et cetera, to inform likelihood. So statistical probability of, so it's like, if you flip a coin a certain number of times, using the likelihood that you flipped it 4 times and it's come up heads every time, it's going to become increasingly likely, just because of the universe and what we know, right, about that you're going to get the reverse effect, the reverse results soon. So that's kind of how it's using that information. And when you get to a certain number of negative outcomes, for instance, the likelihood that it could ever meet the predetermined endpoint of, being positive, becomes so much less likely. So then you can start to move away from that, or even kill that arm, or just start to introduce more a bias of randomization towards the arm that seems to be more effective. And you're just more rapidly trying to address the question. So on a very kind of obviously third grade explanation level, that's kind of what you're doing. And what the statistics are and how often you look and how much you change, those are all the obviously statistical intricacies that you really need a high level statistician to explain. But that's kind of the principle of how it works.
Thank you. That's a great answer. I appreciate it.
Yep.
Dr. Lowy, we're running kind of short on time here, but just for this group here, how does a young surgeon get involved in clinical trials, particularly for people that aren't at a really big place that has a massive clinical trials apparatus? Do you volunteer for committees? Do you need to go to a course like the Vail course? Do you sign up to be PI for your site? Where do you even start? Because this is very daunting for many people.
Yeah, so I would say yes to all of what you just said. So first of all, you want to get as much training as possible. So anytime there is a course or a webinar or anything like this, to participate, obviously. If you have, if you're at a center that runs clinical trials, obviously there is a clinical trials office or infrastructure. And so obviously making yourself known to the folks who are involved, will allow you to get involved in the infrastructure at your institution. But, on the national level, really, I would be I'm a huge advocate for participating in the NCTN groups. The NCTN groups' mission is to train young investigators to lead trials. Our mission always is, when we're creating trials, is to look for young people to lead them. It's not for looking for me and Eileen to lead them. Our success is, we're successful if young folks are leading these trials, and it's our job to mentor them. And each of the NCTN groups have young investigator development tracks. They have courses that teach you, how do I become a PI of a Swog trial? How do I become a PI of an alliance trial? And obviously you want to go to the meetings. Showing up is critical. Participating is critical. And so if you're lucky enough to be able to go to the Vail course, that's an outstanding, course. There are other courses. I think the American College of Surgeons now has some courses about clinical trials, and as do other societies, obviously like AHPBA. So I think you can't ever hear the information enough. If there are obviously senior folks at your place, if you're at Emory, you want to latch yourself on to Shisher Maitel and let him mentor you, obviously, we often mentor people from afar. They don't, I've talked to many folks from other institutions who have questions. I think people are obviously open to helping you, and use your society meetings to network, to meet people. So all these different ways, are helpful. But I think, the cooperative groups are a great place, because that's really a training ground. Again, the goal of these groups is to train young investigators, and regardless of specialty.
That's actually really excellent.
Can I just add one thing to that, just from everything he said from a personal experience standpoint? So those cooperative groups, I think, are amazing, even if you just go listen. But it's very important to find a mentor and a sponsor, and someone who will bring you into the group, introduce you to the group. And keep in mind that 95% of the people you'll be speaking with are medical oncologists. But even if you just go to the meetings and listen, and that's what I did for the first year, I just listened, and you start picking up on design and things, what makes an appropriate design, things to think about, what are the statistical concerns, and all of that. And it's just an extraordinarily longitudinal educational experience. And again, not everyone on this call is oncology slanted. Not everyone's going to be involved in cooperative groups, but you can still learn from that. But I would encourage you again I'm a huge believer in collaboration and learning from people around you. And the truth is our medical oncology colleagues, this is their bread and butter. They're doing trials, and this is what they're doing in their non-clinical days and not in the clinic days. And even just latching on to medical oncologists in your own institution, you don't have to start with an NCTN trial. You can start with a small single institutional trial. When I was 3 years out, I did a small 20-patient trial here that, it meant nothing, but I went through the process of writing a protocol, enrolled patients, took 5 years to put 20 patients on, but I did it. And I learned from it, and it kind of builds your experience with it. And same thing, with not everything's going to be cooperative or NCI funded. There's drug funding, grant funding, and these are going to be smaller trials, whether it's single institution or 2 or 3 institutions. You learn the regulatory, you learn how multi-institutional stuff works. You learn all the logistics that go into these trials, which you take for granted in an NCTN trial, because it's kind of all covered through the kind of, the funding that happens. So many other ways to really just start increasing your knowledge on this.
Maybe one last thing to add to that. I think that's very well said by Andy and Shashir, that some of the cooperative groups have specific committees, and the Alliance has one for surgery, and Rebecca Snyder and Moaveen Omid lead that. And I think that's, just reach out to any one of us. I think we're all involved. We'd be only too delighted to engage anybody who's interested. It's I agree. I think it has a reputation of being a slow-moving process, but it's a really interesting, informative, educational process, and I think it's something to be very proud to be involved in for oncologists and surgeons and the research community.
Yeah, I can also personally say that another person who should be not unmentioned here is Dr. Ahmad, who, I went up to him when I was literally a first year attending, and, got me involved into Swog. And I think it was the same thing. It took literally a year of even understanding what the vocabulary and definitions of a lot of these trial design concepts were. And over time, and I think a lot of these cooperatives were very, helpful and really want to promote young investigators. So I think for everyone who's on the call who's interested, there's many on this platform that are heavily involved, and would be more than happy to guide you. So let us know, email, and we will also have a session specifically for next year in person about sort of clinical trial as a career. So with that, unless there's any additional comments from Dr. Ahmad or Shishir, we'll just sort of end the session. I know we're already over time, which goes to show how I think successful and really informative the session was. I really want to thank our speakers. This was really a tremendous effort on your end and really successful. So 2 other things to mention, that this is a year-long course. So for those who are interested in getting a certificate, we will be sending out a survey plus a multiple choice exam. And if you pass those, you will get the certificate at the next AHPBA meeting, so in 2023. So we look forward to all your feedback. Please let us know what went well, what we can improve on. There's always room for improvement. And really thank you all for joining this late evening.
Thank you, Eileen. Thank you, Andy.
Thank you.
All the best.